Hello everybody at today's afternoon session of the Keynote lecture, and the moderator of today's session is Mr. Alain Chatelain. Alain, please, floor is yours.
Thank you. Dear participants, it's a pleasure for me to welcome you for this Fondation Paralysis Cerebrale lecture. Let me first thank the Poznan organizing committee and the technical team who made this conference possible. Fondation Paralysis Cerebrale is one of the very rare foundations in Europe dedicated to research into cerebral palsy. Our missions are to support research and innovation for children and adults with CP and to improve the quality of care and support to children and adults through dissemination of new knowledge and good practice. We want to be a crossroads where families, researchers, and donors can meet, recognize each other's contribution, and partner for better participation and achievement of goals of children and adults with the condition. In recognition for the important role played by EACD, we sponsor a lecture every year, and the GNC of EACD has selected Professor Andrea Guzzetta to be the 2020 lecturer. Professor Andrea Guzzetta is a professor at the University of Pisa. He is the director of the Infant Neurology section of the IRCC Stella Maris Institute, and of the SMILE, which is the acronym for Stella Maris Infant Lab for Early Intervention. His research focuses early neuroplasticity and very early intervention and infants with brain damage. He has also published on visual development and visual impairment in children. And within the early intervention, the model is strongly centered on early mother-infant relationship and intersubjectivity. He had the opportunity to study and work in various places in all the Hammersmith Hospital with Lily Dubovitz and Eugenio Mercuri, and on the visual side with Janet Atkinson and Oliver Braddock, in Tübingen with Inge Kregelman and Martin Stoltz, in Australia with Roslyn Boyd, and of course with Giovanni Cioni. He is the senior tutor of the General Movement Trust and received in 2012 the Hans Fechtel Award in Developmental Neurology. Please, Professor Guzzetta, the stage is yours, and participants will send questions through the web application before you can respond in the end. Professor Guzzetta.
So, thank you very much, Alain, for your introduction. I hope you can hear me. It's a great pleasure and honor to be presenting at the EACD conference. Really, I wish I could have been there. I love Poland, and Poznan is one of the last big cities I haven't visited yet in Poland. So it would have been really great to be there, but I'm sure there will be opportunities. And also I want to thank the Fondation Paralysis Cerebrale for choosing me for this lecture. So I hope that the screen is shared properly, and I'm sure that people will advise me otherwise. So as Alain was saying, I'll try to basically take you through a journey that I imagined that goes basically from a very specific place in time. That is when my second daughter was 8 weeks old. And you can see here the ultrasound, 8 weeks, of course, postmenstrual age, and she was moving a little bit. Her heartbeat was going, and of course she was growing in the womb. And the journey starts here, and it ends here when she was 60 weeks old, and she was about 5 months postterm, 60 weeks postmenstrual age, and that is exactly at the end of the period of spontaneous movements as we know it. So 8 weeks is when movements in uterus starts, and 60 weeks is when spontaneous general movements are over. in development, and a lot of things have happened between this moment and that moment. And you don't need a scientist to understand that 60 minus 8 is 52, which is exactly 1 year. So that's why I call my presentation the marvelous year. I think this is a marvelous year because there are a lot of things that are happening in such a short time. It will never be like that in the rest of life. And even if we just count neurons, we go from about 20 million to 150 billion at these two time points. So it's really miraculous how things can go well most of the time. And this is going to be what is my lecture about. So I'll try to start by talking about movements and when movements are beginning in utero. And as we said, we know that the embryo starts to move around 7 weeks. We know that because now we have ultrasound, we've been having that for 30 years, and it was possible to do a lot of scientific work thanks to that. So we start moving, and our movings are very simple. So we're bending side to side. This is the beginning of the life of movements. It's the beginning of when the brain becomes active and visible to everyone. But then, of course, things change very rapidly. And we know, for example, that already at 10 weeks, movements are much richer. We have a lot of new repertoires. We have new patterns that are present, and we can see that the movements are more rounded, are more complex. In just a few weeks, a huge change has occurred. And if we want to look at these videos very carefully and try to list all the patterns that we see in the different stages of fetal development, we will realize that is a growing list of patterns. So if we only think that what happens at 10 weeks, we can have general movements, we can have startles, hiccups, isolated limb and hand movements, jaw openings. So we can have already a pretty long list of different patterns because it's very clear to everyone that startles, hiccup have very different features in terms of kinematics and therefore in terms of the neural circuits that are sustaining them. But in just 2 weeks, we can add some more. We're going to have breathing movements, hand to face, finger movements, stretches, and yawns. So very quickly in this journey that we just started, things are changing fast and new patterns are piling up. So the impression is that while the development is going, there is the activation of new central pattern generators that are producing new patterns and new activities of the brain, and that are very much interrelated to each other. So for example, if we take a classic example of a relationship between hiccup and breathing, we know that these two movements don't happen by chance, but they are very much interrelated. So if we, for example, look at a fetus that at 12 weeks is hiccuping, we can see how massive is this movement, how important it is in displacing the baby and probably in squeezing the thorax and producing effects that have been determined and that have been studied, such as the triggering the differentiation of the lung cells or helping nerve pathway findings of the intercostal muscles. So this simple, apparently simple movement is actually changing the structure of the fetus so that a couple of weeks later, when the fetus is a bit more mature and these new central pattern generator of respiratory movements that you can see, I hope, here in this picture are starting, the fetus is structurally ready for that. And so this simple connection between two patterns is also demonstrated by the fact that the first movement, the hiccup, is rapidly reducing in frequency while respiratory movements are increasing. it is clear to everyone that we don't need to breathe at 14 weeks. So these are all expectant phenomena. And there are many others like the maturation of the gastrointestinal tract that is triggered by swallowing and other movements like eye movements. We know how important eye movements are to produce these retinal waves that are essential for the maturation of the visual system. So we could, in a way, imagine the development at this time as something that is occurring week by week, and this consisting in the switching on of central paragenerators, the one we see, and by the time that weeks go, there are new central paragenerators that are activated, and some are switched off, and some are switched on again, and then two are switched off, and so on and so forth. And this is a graphic depiction of what happens in rodents. So this is a rodent model where you can actually follow the activation and the deactivation of these central paragenerators. And so if we imagine this, of course, the journey in the utero becomes a very complex system where timing is so essential and where the activation of central paragenerator is time locked very tightly, otherwise things might go wrong. But of course, this is just a part of the story. So the movements, the central paragenerators are producing movements. But as we have known for many, many years, fetuses can feel these movements. They already have sensation. They have somatosensory receptors starting at 10 weeks gestation. The somatosensory system is the first to develop. And so they are able to receive a feedback every time that they move. And this was clearly studied and demonstrated some years ago in this seminal study by Kazipov in Letters of Nature. He studied rats. This is not a rat, this is a mouse of the lab here in Pisa. And he's, as you can see, he shows all these little twitches. He's at the moment 1 day old and shows these beautiful little twitches continuously. So what Kazipov described and demonstrated is that all these movements, each one of these movements is actually sending, of course, a feedback to the brain. And in 70% of the cases, this feedback is actually producing the spindles that you can see here, this electrical activity in the brain that you can see here. So this is an enlargement of this tracing, and you can see here that the movement starts here before the brain activity is detected, and so is actually triggering this transient activity. It is not all transient activity is not connected to a movement. About 70% is, as I was saying, 30% is natural, is spontaneous. But a lot of the activity in the brain is actually triggered at this time by the sensory feedback of the movement. And this activity is distributed in a topographic manner following the more external areas of the motor cortex that are connected to the forelimb, as you can see here, and the more internal connected to the hindlimb. So it's somatotopically distributed. And what is also interesting in this study is that if you cut the spinal cord, so you remove the connection of the muscles that are moving to the brain, to the nervous system, you cut out about, as I said, 70% of these transients. So really, this study was then replicated many times. We know much more about this topic. And we know, for example, that this is also true for humans. So if you take a little 32-week-old baby and you measure the EEG activity, you will find that in many cases, the hand movements are actually preceding the activation of the transient activity recorded at the scalp. And again, it is also in this case somatotopically distributed. You can see here the activation when the hand is involved, the activation when the foot is involved, that is much more medial and homunculus. So every movement we make in utero is actually producing an effect in the brain, is actually shaping the brain in a way that is changing the connectivity and maturation. So we could imagine that development in the womb goes a little bit like this. You have the stimulation coming from the central paragenerator that goes back in terms of sensory function, and it is actually changing and shaping the brain by this sensory motor loop, and so on and so forth. So again, the activation is changing the brain, and then the brain is producing new movements that are more complex, more mature, that produce new, more complex sensory feedback. This is, of course, an oversimplification. It's just looking at one aspect, but it's probably a very important one. So this hypothesis is somehow confirmed if we think of studies that have investigated the brain of preterm babies, so those who spend the last months outside of the womb, and they have tested their connectivity, and they have found, for example, like in the study by Thompson in last year, that the connectivity is disrupted or is different, let's say, in a way that is correlated with the severity of prematurity. So the difference between a brain of a term baby with the brain of a preterm reaching term is correlated with the degree of prematurity, suggesting that this difference is related to how much time was spent in a different environment. And this, of course, makes a lot of sense. So we can imagine how important it is then to have this complex machinery work in the right way. And actually doing animal model studies, it was possible to manipulate this machinery. And basically in this beautiful review of Lumen 2016, you can see how studies are actually showing how manipulating the spontaneous activity of the brain during this critical period is actually affecting all the different aspects of brain maturation, with no exception. But we're still at 10 weeks. So we were looking at these ultrasounds of a 10-week-old baby, fetus, sorry, actually going from embryo to fetus. And but a lot of things still have to happen during pregnancy. For example, something that is obvious, it's been demonstrated and shown by several authors, is the fact that the general movements are kind of reduced in episodes per hour, while more localized movements start to grow. And after 10 weeks, this is matched with the timeline here. After 10 weeks, these localized movements start to grow and become predominant at around 12, 15 weeks postmenstrual age. So what's happening here? What are these localized movements that are coming and growing and developing? Of course, it's very hard to say. It's very difficult to demonstrate exactly what is triggering those movements. But some aspects of them are clearly demonstrated by research. For example, it seems that some of these movements are triggered by the sensation that they are actually producing in the fetus himself or herself. So basically, it was shown that the fetus from 10 weeks increasingly seems to prefer those parts that are richly innervating, suggesting that, of course, they are able to progressively select the movements. And this is a series of pictures from the very beautiful book by Sandra Piontelli on fetal movements, showing how the hand is gradually going back following the growing and spreading innervation of the scalp. Also, a few weeks later, fetus start touching their eyelids very much. So this type of movement starts to increase. And of course, this needs to be a coordinated movement that is probably producing also some effect at a visual level, like phosphenes, or certainly at the somatosensory level. So the beginning of this increase of more localized movements has been interpreted as something that was called primary sensory motor intentionality, borrowing the words of De La Food. So this is assuming that these movements need to be prospective and anticipatory, because they include a basic awareness of the proprioceptive and or exteroceptive consequences of action. So basically, in order to be able to consistently repeat that movement, I need to have a prospective motor program, and that is generated by this continuous sampling of information that the fetus does, both from the especially from the proximal environment at the beginning, that trigger consistent patterns of neurofunctional activation every time, and then can be accessed by the fetus in order to be repeated in order to have the same expected and wished consequence. So having this memory of consequences probably in these weeks after the 10th, up to the term age, will allow for refinement of motor plans that of course are shaped by the sensory consequences of themselves. The fetus will increasingly be able to select the preferred motor plans based on the anticipation of the sensory consequences, and also can start activating anticipatory behavior in chain motor plans. For example, what has been described as mouth opening when the arm is actually approaching the mouth. So Raceland in 2014 has monitored fetuses, trying to understand what was the chance of and the frequency of them touching the different parts of the face. And you can see that there is a development of these patterns. So there are developmental maturation of each one of these abilities. And for example, touching the upper part of the face decreases significantly, while touching mouth and the lower part of the face increases. But what is very interesting also in this study is that the movements of the opening of the mouth, of the movements of the mouth that are followed by touch, so those that are anticipatory, the mouth opening before the hands touches it, are increasing. And so he claimed this, Schick actually claimed this is a possible evidence of this anticipatory nature of these movements. And of course, this requires very important skills already at this time. But other authors has explored the nature of these fetal movements and have shown, for example, that as early as 22 weeks, a singleton is able to differentiate the kinematics of movings touching the mouth or moving touching the eye. There's no difference up until 18 weeks, but at 22 weeks, this difference is huge. So there is a much stronger deceleration when the movement is planned to touch the eye as opposed to the mouth. Probably because in these sampling inputs of and this experience that the fetus has so far has shown him or her that touching the eye is giving a different sensation, probably too strong sensation to be touched too abruptly. So the fetus is able to differentiate these type of movements. And the same authors have shown in another paper a very interesting aspect of this story. And that is that when you introduce another element in the picture that is a twin, what happens is that the movements towards the twin are of the very soft mode, the gentle mode. So in this study, Castiel and co-workers showed that when you are 14 or 18 weeks and you have a twin in the womb, you make a very, very big deceleration before touching your neighbor in the uterus. While when you have to touch the eye or the mouth or the wall of the womb, you are much less delicate. So they have interpreted this in a very fascinating way. Actually, they call this paper Wired to be Social. So are we wired to be social already at this early time in development? Are we wired to be social when we are still in utero? And well, I strongly believe that during the fetal life, these functional networks that are developed, that we were talking about, that are selected and refined to respond to any stimulus, as we showed before, also have to respond to social stimuli. And having a twin in the utero for 40 weeks is certainly an important social stimuli to some extent. But there is evidence in other studies showing that there are other social stimulus that even when you're still a fetus are able to induce a reaction in the fetus that is showing some maturity in social skills, if you wish. And that is when any communicative biological movement producing either visual or also auditory stimulus is produced. So, for example, when we watch someone moving, or when we hear the movements, the sound produced by a biological movement, like, for example, the voice. And of course, this is a subject that has been extensively studied in monkeys and in adults in that field that is called the mirror neuron system area of science that is very, very developed at the moment. But there are some examples, as I said, of this happening in utero. For example, this is what Pier Ferrari has shown some years ago, just a few years ago, asking mothers to produce la sounds while he was using an ultrasound to see the movements of the mouth. And he did this in 25 weeks fetuses, finding that the control conditions, one was opening of the mouth by the mother without producing any sound, which of course didn't trigger any mouth openings in the fetus. And the other one with all other stimuli like chewing or yawning that were asked, the mother was asked to produce. So when mom was producing these la 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 movements, there was a significantly higher chance to have mouth openings in the fetus. And the interpretation of this was that fetuses have developed some rudimentary forms of motor resonance, which involve the capacity to activate motor representations similar to that of the model, in this case, the mother. And if this is the case, there's no other way to develop this rudimentary form of motor resonance by moving, by producing these movements that are known to create a somatotopic and map of in the fetal brain of what is where in the body of the fetus. Another example that is also quite recent, even more recent, this is a paper that was published 3 years ago, is about vision. Again, the fetuses were tested with ultrasounds and were tested by understanding their movements either towards or away from a stimulus. And the stimulus was three lights that were produced either in this shape, that is similar to a face, or an inverted face shape. And what these authors describe is that at 34 weeks, you have significantly more movements towards the stimulus when it's face-like, as opposed to when it's inverted face-like. And of course, if this is confirmed, this shows that the baby, the fetus, already needs to have a representation of the face that, again, cannot come other than from his own movements and learning from the sensory feedback of his own movements for about 30 weeks at this time, that he has experienced for 30 weeks. And the authors conclude that postnatal experience is not required for preference to engage with a face-like stimulus when contrasted with the same stimulus presented in an inverted configuration. Of course, this is all speculation. We need more studies to understand what happens in the fetus in this respect. But what we know is that the fetus appear to be prepared for postnatal social interaction when he is born. And this is shown by many, many studies in human neonatal imitation that have been studied and just confirmed, but then finally definitely confirmed by the a huge amount of research that is out there. This is the famous video of Pierre Ferrari with his 2-3 day old monkey that is trying to make him or her move the mouth when he's moving his mouth. He will start in a second, and the little monkey will follow. He studied extensively the imitation in newborn monkeys, and he's shown some very interesting facts. And, especially, he showed what are the consequences of this. He showed, for example, that the monkeys that are better in imitating, they're also better in recognizing human social partners. They're better in increasing visual attention to the phases of human. They have more visual attention to the human social partners. They're better at remembering gestures and initiating their social interactions after a delay. And they're also better in reaching and grasping later on in development. So it seems that the skill of imitation is kind of a useful skill for development and certainly for interaction. And studying infants, Lynn Murray showed very clearly this. So she showed in a paper of 2016 by looking at diets in a natural setting that infant behavior is characterized week by week. This is 1, 3, 5, 7, 9 weeks of life. So these are the first 2 months of life. By a basic stability with a little bit of a reduction of non-social movements of the mouth, but a steep increase of social behavior. And social behavior composed of pre-speech, vocalizations, and smiles. And at the same time, maternal response was changing. Again, the neutral marking, negative responses were not really changed over time. But what was changing is the ability of the mother to mirror and to give a positive marking to the infant behavior. And this is very important to build the relationship and the interaction, is shown by another result of this study, and that is that the maternal responses are actually correlating with the social abilities of the infant at the next appointment. So the more I'm able to mirror my son or daughter signals and to mark them positively, the better it will be his or her social skill in a week, in 2 weeks' time in this case. They concluded saying that the preparedness of the infant's perceptual system to attend to particular social stimuli is matched by the propensity of parents to deploy specific responses to different infant behaviors. Infants appear to capitalize on the limited corpus of these responses to develop their social expressiveness. There's no need for a huge amount of contingent interaction between a mother and infant to help the baby to develop their social skills. And this probably also suggests that there is an innate component of it that is not due to associative learning after birth. So going back to where we started, this was a very long journey, just a few minutes, but where I try to describe what we think, what science thinks happens between this time and this time. So between the beginning of movements and the building of a very mature interactivity skills by mom and baby. Of course, this is the end of this journey, but for this little baby it will be the beginning of a bigger journey from now onwards for an n number of years and months. But as a clinician, of course, and being also presenting at the EACD, the question is, how is this relevant for our little ones, for our cases? And of course, I think it's obvious to everyone how this is hugely relevant. But I would like to end by just mentioning two things, showing you two examples of why this is relevant. And in particular, what I want to focus on is on the fact that when a baby is born and has brain damage or has developmental difficulties, we have to understand and keep in mind that he had gone already, he had a long journey before that moment. So our goal must be not only to make him improve in the future, to make him better, but also to be sure that he doesn't lose all the knowledge he has learned in this journey. And these are the two examples that make me think of this aspect. One is seeing a baby that has cramp-synchronized pattern. And we know that this is a very discomfortful pattern. This is a pattern that is associated to cerebral palsy. And we can imagine these babies moving in such a very complicated and painful and a natural way for many, many hours every day. And so if we think of this, the journey this baby has done so far, we should try to make sure that these movements are not destroying what he has constructed in 9 months of life in the utero. And so that's why we do neurodevelopmental care. That's why we do massage. That's why we do nesting and kangaroo care. But also, that's probably why these authors recently tried to play a new approach that they call movement imitation therapy, in which they try to reproduce more natural, more smooth and complex movements passively in this infant. And they manage in 4 cases to make the babies grow out of the cramp-synchronized into a fidgety and normal fidgety. Of course, it's very fascinating, very preliminary data, but interesting to reflect on those. And the other example, this is my last slide, is another example, not based on movement, but based on relationship. This is a baby that is 12 weeks old and has brain damage, severe CVI, and is trying in this interaction with mom to connect with mom. And you can see how hard it is for this baby to connect and for mom to be able to connect with her. You can see that she tries very hard, but then it is enough to facilitate this encounter to reawaken some skills that this baby certainly has and shouldn't lose because of the lack of vision. So just by making the phase of mom more salient, something miraculous happens. The baby starts to be, begins to be more calm and more concentrated on mom. In a minute, you will see how it stops moving and looks at mom and finally starts smiling, showing that these skills are there and shouldn't be lost because of our delay in taking care of them. And we assessed, of course, with a macroanalytic approach, the interaction showing that with facilitation, the attention of mom positive movements are much significant, much bigger. It's one case, but it's much bigger than without the facilitation. So this was my last slide, and of course I want to thank the amazing collaborators I'm so lucky to have in the Stella Madison Fund Lab for Early Intervention. Here is the COVID time. We are wearing masks, but I can assure you that the Smile Lab has smiles from the masks all over. And of course, also some relevant collaborators relevant for this talk, Limma Samsa Anathalo, Pierre Ferrari, and of course, my mentor of 25 years, Professor Choni, and of course, all our funders. Without them, of course, we couldn't do any of what I showed you. So thank you very much. Thanks.
Thank you very much, Professor Guzzetta, for this passionate talk and very interesting presentation. I'm just having a look at the question. I'm not sure whether you can see them, but maybe can I start with the first one? Can we say that the movement patterns of children's cerebral palsy with premature and term may be different even if they have the same clinical type?
Well, yeah, so the movement patterns of children with cerebral palsy, I'm not sure I understand the question. So the pattern of those that were born premature and those that were born at term may be different if they have the same clinical type. Yeah, well, sure. The impact of the damage that is actually producing cerebral palsy down the line is also producing different patterns in movement type, especially of course the deviation from the natural development of movements has a different timing. In the preterm baby, we have an insult that occurs in an earlier time. So usually these preterm babies have the ones who develop cerebral palsy have a repertoire that is very impoverished, and then have this cramping later on that appears. And then develop cerebral palsy. In the case of a term baby, it might be different. It might be different. It might be that they don't have a cramp synchronized, although they can still have it. But I think that it would be more sensible to say that potentially these two pathways are different also in terms of movements, the one of a preterm and a term. Then I have a question by Naomi Dale, who says, are
there any preventive compensatory? Sorry, go ahead.
Are there any preventative compensatory inputs we can put in for babies who lack vision or motor movements? Well, I think with these last two examples, what I was trying to show is that probably we have ways to spare what is the result, the huge result of the part of journey that these children have already made. And so we know, and we share this with Naomi, this idea that helping babies to make sense of the environment around them by facilitating the visual environment, by making it more salient, by directing their attention to it, by making it more focused. This can certainly promote development. And the example I showed about the movement is also an attempt, I think, to do something similar. So to try to give back to the brain of these very sick babies a more typical, more natural, more normal, if you may, somatosensory feedback by making them experience nice and smooth movements. So yeah, I think it's a very interesting topic and passion we all share here. And then I have another question. Has any sensory enrichment shown a reproducible effect on development when applied prenatally? For example, music. Sensory enrichment has shown to change this set of knowledge of the baby. For example, babies that are exposed to music as fetuses remember the type of music they heard of. So they heard during the pregnancy. So that means that they have a different brain that is more sensitive to that particular music. So in broad terms, sure, any enrichment during fetal life is actually shaping the brain in a different way, and that applies also for, of course, sound and music. Are GMs before 2 months old age valuable for estimating more outcome in infants? Well, actually, GMs are currently considered in the recently published guidelines, international guidelines in 2017 by Iona Novak, are considered the best tool actually before 5 months, and also before, I mean, in that time period. If we think of babies that are have not reached fidgety age, so before 2 months, as the question asks, at that time, the GMs are not as predictive as later on, because you have a number of false positives. These are all those babies that have poor repertoire that actually will develop normally. So at that time, you probably need to rely on other pieces of information in those cases, unless you have a severely impaired cramp synchronized baby, and then or otherwise wait, as we often do, the second, third month as a turning point is in those difficult cases in which we have not cleared the outcome before then. And Eric says,
I think we have reached.
Oh, yeah, okay.
Andrea, I think we have reached the
sorry, yeah,
time limit. I don't know.
Yeah, so, I'll type the
unless we are careful. Yeah, if you can type your answer, but I think we have to give back the talk to the organizer for the next presentation.
Absolutely.
Thank you very much anyway, Andrea, and we should have other opportunities to hear your important work. Thank you very much.
Thank you.